Welcome to the second episode of ISB Presents Curate This, a podcast that pulls back the veil and shows how biocuration is done in practice. I'm Charlie Hoyt, a member of the ISB's executive committee, and today I'm talking with Dr. Victoria Nugnes, an Argentinian biocurator and postdoc at the University of Padua. Victoria is the lead curator of the DisProt database and works on the annotation of intrinsically disordered proteins. So the first question is, what is the name of the database that you work on?
Our resource is called DisProt. It's the database of intrinsically disordered proteins, and recently has been selected as one of the Elixir deposition databases. So we are really happy about that.
And how old is this database?
It was established in 2005, so it's been around for more than 2 decades.
Wow. And so for those 2 decades, has this resource been developed just by your group or in a combination of your group and outsiders? Or what does the curation team look like?
Well, it began being handled by another group in the US, but then we have been hosting it for almost like 15 years now. And it started like as a small expert-driven effort, but today is much more like community-supported resource with professional curators and also contributors from a broader IDP community.
IDP is intrinsically disordered proteins.
Yes, sorry.
Okay. So how many people are working on this? Like how many full-time curators, how many developers, and then like how big is this community of people who participate?
If we just consider like the professional core, it's like mostly professional curators and also developers. If we consider also the PI, let's say it's like about 6 persons, 6 people. And we also collaborate with different international researchers. Let's say right now we have like about 20 community curators that contribute in common basis and usual basis for the research as well.
All right. That's a really nice community. So more specifically, what kind of information is stored in DisProt?
What we do, we store information about proteins, particularly proteins that have disordered regions or are completely disordered. These are proteins that have flexibility and don't have like a fixed 3D structure. So what we do is what we collect experimentally validated information about these proteins, whether they are related to the structural states of these regions or proteins or the functions. We collect experimental evidence that was used for the assessment and the region boundaries that are being assessed by other researchers.
And just a little bit of biology for those who aren't familiar with disordered regions of proteins. What is the impact or why is it important that proteins have disordered regions?
Well, disorder is a feature for many of the proteins, but sometimes it's also pathological. So it is related to different neurodevelopmental disorders or even cancer. When there are some mutations, one protein that wasn't disordered before could develop a region that increases its flexibility and gets disorder and has a pathological phenotype. But then there's other cases where proteins and disorder regions are important for the function of the protein, for the biological function of the protein, for example, for stress response proteins or for viruses to avoid immunological recognition from the host. They are also very related to proteins that have a high concentration of disorder regions.
And you said that the proteins can develop disorders. So is this typically a regular protein that becomes disordered when it's not supposed to be in pathological sense? Or is it that a certain mutation will cause a region to be disordered when it's not supposed to be? Or both?
Both, actually. And sometimes it's regions that are disordered when they're in a healthy state, let's say. But then these disorder regions, since they're too flexible, they try to condensate and then collapse, forming like amyloid formation or fibrils that prevent the normal functionality of the protein and then become pathological.
Okay, good. I think we're all on the same page for the importance of disordered regions and quantifying them. While you're building DisProt, does your group reuse any data standards and does it develop any of its own data standards?
We do both. We reuse and try to make the most about for external standards like gene ontology. We use gene ontology for the definition of functions, or also ECO, evidence and conclusion ontology, to annotate the techniques that are used by experimentalists to assess these features, and some control vocabularies like from the UPO PSI initiative. But we also have our own. We have intrinsically disordered protein ontologies, which is IDPO, which is very related to our scope and to define the different kinds of disorder or functionalities that are very, very much related to the disorder nature. And we also have some guidelines related to the description of the experiment that was used for the disorder experiment. That it's called MIADE.
Okay. So taking a step back, I want to know about the impact of your database. So what kind of users use your database or could use your database and why? And also, how do you assess this?
Well, I think it's a variety of users of the data. There are some that are working on a particular protein that they know it's disorder or a particular disease, let's say. So they go into DisProt to find out what's been assessed regarding the structure or the functionalities of this set protein. Or there are other like structural biologists, like more generalistic, or computational biologists, bioinformaticians that are developing like predictors to be able to assess disorder regions in proteins. So what they do is they collect the information that we have on DisProt to be considered as like the ground truth to train these predictors. We assess these different use cases based on citations, of course, but also in the lab, we also run CAID, which is the critical assessment of disorder predictors, in which predictors are submitted and are assessed on whether how much they relate or can predict disorder in newly unreleased data from DisProt. So we do like a categorization or different challenges between the different predictors that are submitted.
Mm-hmm. And so going back to something you said already, the broader impact of your resource within the basic and translational research space in biomedicine, you mentioned that protein disorder is a key part of some diseases, especially ones with the formation of fibrils or aggregation of clumps of proteins like amyloid. I think this is a big deal in neurodegenerative disease like Alzheimer's disease. So is there anything else like that besides that part that you think that this is interesting and has a broader impact on the research landscape?
Well, yeah, exactly as you were saying, there's not just like the basic research focus, there's also like the translational efforts. And disorder is also being used as or studied to understand different biomarkers or even for drug discovery. There are a lot of new studies about disorder peptides being used as drugs because they are very, very, they can bind to multiple partners. So they're being used or studied as different drugs.
So having like a global understanding of the landscape of disorder proteins can inform drug discovery where they want to use this physical feature for translational purposes. So the last question before we move on to the demo is, how do you onboard new curators to your resource?
New curators have to go through training. It's the same for internal contributors or for external contributors as well. We communicate via Slack. We have regular meetings, like 2 months, to discuss what's new in the database or any new developments in the web page and to discuss any kind of questions or struggles that they have been regarding the curation of new information.
Okay, then let's do it. Let's get to the live demo part. So the next segment, we're going to actually look at the process of curating information about intrinsically disordered proteins, starting with a paper from PubMed. And I'm going to leave it to Victoria to explain what she's doing and her thought process as a curator and how she trains other curators to do this. I might bump in with some questions as we go, but it's up to you. Go ahead.
Thanks, Tali. Well, when we are looking into information, we're looking to find information to curate and to add into DisProt. We normally can perform publication or scientific publications searches by looking into like keywords like intrinsically disorder, for example, a disorder, flexible, or the different techniques that are used for the assessment of these particular protein regions. So let's say that we found this publication. The title is Structure and Dynamics of an Intrinsically Disordered Protein Region that Partially Falls Upon Binding by Chemical Exchange NMR. So this seems right into DisProt alley. Okay, so the first step is to make sure that we didn't curate the publication already. So the first thing is to take the PubMed ID and we go into DisProt. I am logged in as the curator and we can use the browser to search for the reference identifier and just type the PubMed ID there and we'll see that we cannot find any entries related to this publication. So now, once that's checked, we can go into the publication and look for which kind of information there is. So the first thing that I do is to, of course, read a bit about the abstract. In this case, I pre-highlighted these sentences. Here, we introduce an approach based on monitoring chemical exchange by NMR to investigate the state of an IDR that falls upon binding through the observation of the free state of the protein. Okay, we know what they've done. We apply the method to investigate interaction between a disorder C-terminal region of Artemis and the DNA binding domain of ligase 4. So it's definitely about something that is a protein that is disorder. So we can go ahead and curate this publication or look into more deeply. What I use actually is an extension from Chrome, which is called Highlight This, in which I can load different word lists, and they can highlight in different colors the words that are related to different features that we curate in DisProt. So for example, we have in orange words that relate to disorder, red words that relate to methods that are usually used for the IDRs or IDPs, intrinsically disorder proteins assessment, and in purple, words that are related to transitions that sometimes could happen between disorder states and order or folded states within the proteins.
Does this sort of mirror the construction of the intrinsically disordered protein ontology?
It's very similar. For the terms that are in orange, definitely. For other things, and not so much because we already relate to other ontologies. But yeah, this is very much like that. So this helps me to get like idea of what it's being discussed in the different sections of the publication. What I like to do first is to go to the experimental section before, just to make sure that they are implementing experiments and not just prediction or inferences regarding the disorder state of the protein. So when I come here, of course, there's like the section about the protein and the peptide production. And then I see isothermal titration calorimetry, NMR resonance assignments, some other assessment regarding the NMR, and some molecular dynamics. But okay, because the first two are related to hands-on experimental approaches, like wet lab approaches. So this is good for me to go ahead and just check what it's said in the results section. Of course, the introduction doesn't make sense that we read it right now because it's a demonstration, but of course, the introduction might provide a bunch of information about the protein and help us understand the publication. But for the sake of this demonstration, we are jumping to the results. So they said that they have been investigated interaction between a disordered C-terminal region of the protein Artemis and the DNA binding domain of ligase 4. Artemis contains a catalytic core comprised by beta-lactamase, Cas domains, as well as the long C-terminal region that is predicted to be disordered. We need to be very careful about these kinds of statements because on the first glance, we might think that this is a curatable information or useful information for DisProt. But again, this is more of an inference by the author and more related to predictions, and we are not getting any kind of information about how this was assessed. So we keep on reading and try to find something that relates to an experimental approach. And then we have ligase 4 binding site of Artemis extends beyond residues 485, 495. And we see that they obtain a nearly complete NMR assignment of the backbone resonances of Artemis. The narrow dispersion of the amide chemical shift in the HSQC spectrums show that Artemis is highly disordered in solution, as confirmed by secondary structure propensities based on chemical shift. This is just what we needed. This is something that we can add into DisProt because in DisProt, we rely on these statements from the publication to do our curation. So let's say this is very good for us. Let's highlight it. And then let's say, okay, we are going to be describing Artemis protein. Okay, let's identify this protein. We know it's Artemis, but we don't know which organism it is. So let's go back to the experimental section and see what they've done. The expression of the vector with Artemis, and we know which region of the protein, 480 to 575, encode a fusion protein with amino-terminal GST domain and protease cleavage, and the Artemis 814 575 region. So they go on and on about the peptide and how it was produced, but we don't have the information right here about which organism or what this actual sequence of the protein is. So what we do in these cases is we check the supporting information. In this case, they have the supporting information as a PDF. So we assess the PDF, and between all the information that we have in the supporting information, we have the full materials and methods. And we scroll down and we see that the gene encoding the region of human Artemis was cloned by. So now we know which protein it is.
So I noticed that, that sentence said it was expressed in E. coli. And so for us who haven't been in the biology lab for a while, I guess this is a gotcha for curators, right? This doesn't, because it's expressed in E. coli, it doesn't necessarily mean it's an E. coli protein.
For sure. That's exactly it.
Okay, good.
So what we do then is we go into UniProt, and we Artemis human, and we look the results that we can have. So this is the first result that we get. We should do some checks. If the publication has any figure or anything that discloses the sequence, we double check with the sequence that is deposited in the UniProt entry. If not, we just try to make sure I'll double check as much as we can. We know that the region that they are cloning or using is from the 480 till the 575. This falls under what's possible for this sequence and this protein. So we are going to use this UniProt accession to identify the protein. So once we found the publication with interesting information and that we want to curate, and we identify properly the protein, we go into DisProt and we try to create a new entry. We paste the UniProt accession number, and in this case, this protein already exists in DisProt, but this could be a protein that did not exist in DisProt before. Since the publication was not curated, we are welcome to add more information. So let's hit the editing existing entry. And this is how a DisProt entry looks like. We have the general information about the protein, which is the tree from UniProt as well, like the organism, the gene, the sequence length. We then have the feature viewer where everything that we are going to be annotating is going to be displayed here. And then we have some external tracks like interpro, missing residues, and AlphaFold that give us an idea of what's already described for the protein, whether in terms of domains, or in the case for AlphaFold, a predicted disorder region. So what we see as in red here in this graphical representation of the protein, this stands for the length of the protein and the position of the different amino acids. This longer part in red stands for the region of the C-terminal that is predicted to be disordered. So going straight to the curation, this is our curation interface where we are going to be adding all the information. We are going to add the PubMed ID regarding to the reference that we have been checking, the Ecoterm or the technique that was used by the author for the assessment, the boundaries of the region that we are describing, the start and the end position, the thing that we are describing, the aspect that we are describing, cross-references if we have cross-references to different databases like experimental deposition bases or stuff like that, and statements from the publication that support what is being annotated. So we have the reference identifier. As we know, we have the PubMed ID. We're going to add it here. We're going to see that DisProt retrieves the name of the publication for a double check as well. And then we can type in the method that was being used. If we type nuclear magnetic, this is what, let's go back to the thing that we wanted to add. In the results section, we obtain a nearly complete NMR assignment of the backbone resonances. Chemical shift of the HSQC spectrum shows the Artemis. So this is an NMR. We can type in again, nuclear magnetic. We're going to get all the terms that are related to this technique. So we have a nuclear magnetic resonance spectroscopy evidence, which is a more general term. We try to be as specific as we can. So we can also just type nuclear to get heteronuclear single quantum coherence spectroscopy, which is HSQC experiment. And now we can add the information about the region that we are describing. As the author says, this is the region from 480 to 575. So 480, 575. The aspect that we are annotating is a structural state of this region, which is related to a disorder state. And then we usually go and try to look for references. Since this is an NMR, we go to the different resources that store this kind of information, like PDB, for example. And what I do is type the PubMed ID to see if there anything deposited there related to this publication. There's nothing in PDB. I also check DMRB, which is a database for researchers to deposit their NMR experiments. And I do the same thing. I type in the publication. We can also check for the UniProt accession number, but I've already checked. And for the purposes of this demonstration, I'm not going to go through it. And there's no cross-references, so we can leave it empty. And then for the statements, we are going to select the source of the statements, in this case, in which section this is being described. In this case, is the results section. And then what we do is we paste exactly as we see it in the publication, the sentences that are found there and that supports that this region is disordered. We save it, evidence is submitted successfully, and now we can see the evidence that we just added in DisProt with all the information.
That's it. We did it. That's a minimum viable curation for DisProt. But we're not done, right? There's actually something else that you might want to do when you're in a document like this.
What we try to do in DisProt is to do a completeness of the curation task, whether it's completing the whole curation of the whole available information for the protein, or at least the available almost information that we can from the publication. So we are not going to be doing that today because of the time constraints, but I wanted to also show how we also collect other kinds of information like functions. So the publication keeps on talking about the NMR findings, but then they also do a different kind of experiment with another protein. They say that an NMR titration shows that extent of interaction with Artemis with the DNA binding domain of ligase 4. The intensities of the cross peaks of our Artemis residues 485 and 499 decreased substantially upon the addition of substoichiometric amounts of unlabeled ligase 4, which is a marker for slow intermediate chemical exchange of the chemical shift timescale. For those of us that are involved in studying intrinsic disorder and this kind of experiment, and if we were to keep on reading the publication, we will see that these signals are for the bound form from Artemis, because Artemis is binding with a ligase 4 DNA binding domain. So we are going to be trying to add this information into DisProt. So let's take this as a statement, copy, we delete everything that we have just added because we are not going to be curating this.
Title drop. That's the podcast's name.
We paste the statement here. And what we are curating right now is rather a function that we use gene ontology terms to define because it's more of an active function. Is protein binding.
Is there something more specific than protein binding in Go that would be valuable, or does that not exist?
Yes, they have many things. What we do in that case is check AmiGo. We check the whole branch of protein binding, and maybe it's enzymatic enzyme binding or domain, certain domain binding. In this case, we try to relate to the protein ligase binding, but since there's not ligase binding, we keep it as protein binding. We do try to be as specific as we can when still providing the information that is key, in a sense. So protein binding, but then we can identify further this protein. How we do that with UniProt accession number. So let's go back to the publication and try to understand which ligase is this protein. Let's go to experimental section. And in this case, the authors do say that the DNA binding domain of the human ligase 4. In this case, it is reported in the experimental section that is the human ligase 4. So ligase 4, human.
You lost an E.
DNA ligase 4. And then we have the identifier. We come here and we paste the identifier right here. We can also identify further which region of this partner is the one that is binding, and we know is from 1 to 240. So 1 to 140. In this case, this is related to an NMR experiment, magnetic. And the region that we are trying to add here is which region of the protein that we are annotating is the one that it's binding. So which region of the Artemis protein is the one that it's binding. Let's go back to the results to see what the author said. The cross peaks for Artemis residues are from 485 to 499.
So this is interesting because it's a subset of the whole region that they were quantifying earlier.
Exactly. We try to be as specific in every case that we have. Like we could annotate this as the whole disorder region, but to be honest, what the authors found is that this region is within the disorder region. So we want to be very specific on that because this could be related to a motif or some specific residues that are part of that region. So by doing this, we have all the information that is required from us. We click on save, and we now see that we have our own function annotation done in DisProt regarding the Artemis binding with the ligands 4. We can also see that in the feature viewer now, we have the representation of the different regions that we have just annotated, the disorder region and the binding region that correlates to this particular segment of the protein. And that's it. Of course, there's more information in the publication, but we're not going to be checking that right now.
All right, so we successfully curated a record on the human Artemis protein and its DNA binding activity, or DNA ligase binding activity. So now we have a good feel of what's the minimum viable curation. We've seen what the curation interface looks like for DisProt. So the follow-up questions after we look at curation is what happens next? Do you have a second curator check for inter-curator agreement? Does the results of the curation you just did, do they end up on the website directly, or did they come in releases later?
What curators add, they're going to like a separate collection, let's say, that user cannot access, but we that are admins or curator of the database, we can see. What we do is they go through a validation process. So one of our experienced reviewers or experienced curators double checks all the information. So what we do, even more so for newcomers or people that are just starting to curate, we check the information that was added and we provide feedback. If we need them to do any kind of modifications or be more specific or whatever, we ask the curator to do these changes. And then once everything is okay, we validate it. So it has like a validation cross check and double check mark, and then it's ready to be released. We do releases every 6 months. So all the information that is validated get to be released on the next date.
Nice.
Release date.
So we had a pretty good experience going through that paper where we found very, very detailed information quickly. Is this like a typical paper, or are there some other challenges that normally pop up during curation that you run into?
Usually we find different cases. There are cases where the publication is very clear on the protein that they're discussing. Like they even maybe they provide a UniProt accession number on the NCBI identifier. Those are like the best case scenario, when you know exactly what they're doing, like which region of the protein they are assessing, if they do mutations, everything from the experimental setup is there. Those are like the best case scenario. In this case, we had to look for the ID or the organism in the supplementary materials, but it was there. There are other cases where it's more tricky. There are cases where they just mention or say like the name of the protein, but they don't tell you exactly which organism it is. And maybe you need to go and find out via different kinds of information. Like you are like private investigator. Like, okay, they're mentioning like this residue is like a histidine, or maybe this is not this protein, this is another protein, or maybe there are figures where you find like the image of a sequence and then you have to cross-check it. These are not very common right now. Probably they're related to more like maybe before UniProt was there, or before they have a like a stand entry or something. But it's usually not the case. We try to look for information that is clear and well explained. So those are the cases that are the most easy to curate.
Okay, so you kind of answered this question already, but like, what would you like authors to do to make papers easier to curate, or not just authors, but also journals and publishers?
Well, yeah, basically this. Be very wary of, try to identify as much as you can, whatever you are doing, whether it's the protein identifier, kind of experiments, if there are modifications, of course, try to avoid vague statements like appears disorder or appears to have something, or if those are related to other people's work, cite them, and if it's related to your work, please also like try to relate to those, because we test this and that. So try to relate to the experimental approaches that were in place. For us, it's very important that are clear distinctions with what is inference or predicted and what's from experimental determination. And of course, also for our case in particular, for us that we are studying disorder is very labile, and it has the context could have a lot of consequences of the disorder status and the functions. So the conditions, temperature, whether there are other proteins in the sample or any kind of things that we need to know to be doing a proper understanding of the results. It's also very, very useful.
And one big question the entire biocuration and more general data stewardship community is thinking about is, do you have an idea where AI has a place in the curation and the maintenance of DisProt?
I think it definitely, yes. I think it has a place. We just need to work with it instead of against it, and try to figure out how it can assist us in our job, and to make the database more reliable and better quality. And for us, what has been most tricky these days is to, while ensuring the quality, keep adding information. And it's very difficult because we want to have more information, more proteins, more information, but we also need to set up a set quality standard. And it's difficult to keep that when you are trying to add more and more information. So AI could assist us in making sure that everything relates to each other, that there are not issues in which is the information that we already have, on top of course, triage services and look for a publication. These days for us, it's very difficult because of all the new techniques that are also arising for prediction or computational studies, for us to find experimentally like wet lab assessment of proteins. So having an AI tool to help us gather this publication and curate it more efficiently, of course, it has a place in this project. We are developing it actually with another group from the Swiss Prot Institute, a triage service, which is called DisTriage, that collects information, tries to collect publication for us to have a more like a speedy process for adding new information in DisProt.
Okay, so that was the show. And as we come to an end, Victoria, do you have anything you want to plug? I know that it was important. You mentioned that DisProt has recently been recognized as part of the Elixir core resources. And so that's like a nice accolade for the resource. We're going to add links to the papers that we curated, the proteins that we looked at, and eventually the record inside DisProt will also link to that. But you said it takes 6 months for these things to come out, so that might be a while. Anything else you want to mention before we go?
Not much, just DisProt is always welcoming new curators. So if you are interested to contribute to this database, you are welcome to join us. I could also add some links there for the training course that we have online, and everybody are welcome to join us and expand on the information of disordered proteins.
Great. Thank you very much, Victoria.
Thank you, Charlie.